Hey guys, welcome to the very first video on what's called Mendelian genetics. Now, I have two goals for this lesson. First off, I want you guys to be able to predict the offspring traits from parent traits, and secondly, students will be able to define homozygous, heterozygous, and dominant and recessive. So, here's some key term review before we move on. First off, gene. Gene is just a molecule of heredity you get from your parents. And if you remember right, these genes are on your DNA. Secondly, the term is chromosome. Hopefully you guys know this one by now. It is a structure inside of the cell that holds DNA. And finally, you have genetics. This is just the study of heredity. It's how traits are passed from one generation to the other. Or maybe a better way to look at it, it's why do I have the same blue eyes that my mom and my dad have? That's what genetics is all about. Now, before we get into the meat and potatoes of genetics, let's first talk about the guy who basically discovered all the rules behind it. There's a guy named Gregor Mendel. Gregor Mendel basically discovered all the rules of genetics. He used an experiment where he essentially bred thousands of peas and he kind of discovered the basic rules. And we'll come back to him later and we're going to spend the next couple of slides going over sort of how he did this and what the general process on all this stuff was. But basically, when you hear the name of Gregor Mendel, you should associate this guy with the discovery of genetics. Now, one other cool thing, or I guess unfortunate thing about this poor guy right here is he did all of this work on genetics with peas, and nobody even knew he existed while he lived. It wasn't until much later on, I believe it because it wasn't really until much later on that people even discovered he had done his work. He had written extensively about all this, but he never really published any of it, which is unfortunate because literally it took almost, I believe, like 40 years after the guy's death before they even found out, hey, this guy basically solved one of the biggest mysteries of life and we had no idea he even existed. So, here's how Gregor Mendel did it. Gregor Mendel took a very basic experiment. He took yellow and green peas and he basically made them have babies. And this is a sample result of what he had. So he took yellow peas and he had green peas and he had them have babies. What he discovered is when he did that all the pea babies were yellow. Well, that was interesting. So he kind of wondered why that was. He goes, because you took the two colors, your instinct would be, okay, yellow and green, that makes yellowish green color. But they didn't come out yellowish green. They came out all yellow. Well, that was a really weird result for Mendel. And that kind of came out the question, well, what's going on? How come there were no green peas after combining the yellow and green peas together? Now, a cute little note here, that when he did this, we have different terms for each one of these generations. So in the parent generation, he took yellow and green and he made them have babies to create a brand new set of genes, in theory. In the F1 generation, that just basically means it's the generation after the parents. So basically, it's the parents' babies. That's the F1 generation. Now, what Gregor Mendel then did is that he made all of these kids, here basically also have babies. So basically, he inbred peas. And what he discovered was something very weird. What he discovered is we made all these groups have babies. He discovered 4 different types of patterns of babies that came out as a result. So for example, when he had this pea right here have babies, he got all yellow peas once again. He didn't get any green this time around, just as if he had taken this one here and had him make babies twice. The second time around, he took this group right here and he made them all have babies together. What he discovered is that 3-to-1 ratio reappeared, where you had 3, 3, 3, 1, 1, 1, and then 1 green. Then he took this group right here and made them have babies, and what he discovered he had 3 yellows and 1 green. So that weird 3-to-1 ratio reappeared. And finally, he took this generation right here and he made them have babies, and he discovered they all came out green. This is a really weird result because what this shows is that whatever's going on is that there's something at play within the genes that is causing it to come out in this weird ratio right here, where you have one set of babies. After you cross here, they're all yellow, which means that whatever was causing this guy to be green is now gone. These guys don't have that green gene anymore. the green gene still seems to exist here, and it still seems to exist here, but it's weak for some reason. That green gene only shows up 1 out of 4 times. And right here, for some odd reason, the green gene is always there. So it seems like the yellow gene has been completely removed from this group. These were cool results for Mendel to get because it really showed that you could actually control how genes were going to be transferred around, and you could actually even begin to predict what animals would have what genes based upon their babies. So this is where the revolution came in. He discovered, based upon this rule book right here, that genes, they come in pairs. So, for example, in peas, there's a yellow gene and there's a green gene, which you can see from here. There's a yellow gene and there's a green gene, and this yellow and green gene seems to be passed along from parent to offspring. But he also discovered that he goes, now peas also have a wrinkled gene and a smooth gene. So you're going to see situations where there are wrinkled and smooth peas. And humans, as an example of the same situation, humans have what's called attached and unattached earlobes. So you can actually see an example here of where this woman's ear is attached to her head, whereas in this man right here, the ear is not attached to the head. So this guy would have an unattached earlobe. And it follows the exact same rules that he discovered right here. Now, he called these little genes, he called these pairs. So when genes come in pairs, he came up with a key term for that. And the key term was called an allele. And it basically says this, is that when a gene comes in different forms, so you have a yellow versus a green allele. If you have a wrinkled versus smooth allele, you have an attached versus unattached allele. So when you see the word allele, all they're saying is that it's one type of possible gene you can have. You can either have a yellow allele or a green allele. You can have a wrinkled allele, you can have a smooth allele. That's basically how the system works. So now we show alleles in a very certain way. So here's an example of how we can do it. Now right here what we have is that we have Y equals yellow and G equals green. And really guys, what letter we use doesn't really matter as long as you tell me what it means. You could say X is green and A is yellow. It doesn't matter. You just have to say what is what. Now, what Gregor Mendel did, going back, if we look at this right here, this is the exact same chart, these two charts. This one right here, though, shows you what the alleles were, and it shows you what actually happened. So, for example, the first one was YY. Well, YY gave yellow, and you got GG, and GG gave yellow. Now, when you had them have babies, well, half, as you know, half comes from mom, half comes from dad. So the parent, the babies here, had a Y and a G. Now, here's where things got interesting, is that what then happened was when you cross these guys together, who will have one for yellow and one for green, and if you remember right, these were all yellow. That means that even though they have a gene for green, these peas aren't being green, they're being yellow for some weird reason. So even though they have half green, half yellow, they're still only showing up as yellow. Now, when he made this group have babies, something really weird happened. He got this weird ratio. He ended up getting 3 yellows and 1 green. Well, now you're going to see what's happened. He goes, oh, so one baby had 2 yellows, one baby was just like the parents, one other group was just like the parents, but one baby got the 2G green, got GG again. And you can tell that by how it works down here, because you got that 3-to-1 ratio, or in this case, they were all green. So what this showed is something is very cool. And it really alludes, which is a fancy way of saying, it kind of hints at something. It hints that certain genes overwrite other genes. And this is where we come into the idea of dominant versus recessive. A dominant gene, a dominant allele, is when you have one allele for a trait, means you have that trait. For example, yellow here is dominant, because if you have just one allele for yellow, you're going to be yellow if you're a pea. Now, there's another key term here called recessive, and that means you have to have both alleles in order to have the trait. What that means is this. In order for a pea to be green, it has to have both big G's. Period. It's that simple. If it doesn't have both G's as its alleles, or both the green allele, it's not going to be green. It's going to be yellow and green. So this is what this means. So a dominant trait just means you have to have one of them in order to have it. So for an example, if you're YY, you're going to be yellow. If you're YG, you are going to be yellow still. If you are, now in humans, attached earlobe is dominant. So if you are AA, that means you are going to have attached earlobe. If you are AU, or I have U here meaning unattached earlobe, that means you're still going to have an attached earlobe. You'll still have the unattached gene, you just won't know it. Now, a recessive, you have to have both. So in order to be green pea, you have to be GG. In order to be unattached, you have to be UU. This is what Mendel discovered, and it's really cool because he really discovered that, hey, as long as you have the genes for it, halas, you're done. If you have both of them, you have a recessive gene. There are dominant genes that seem to take over and overwrite the recessive ones. Now, there's another key term here before I go into how you can calculate this stuff. Now, there's two key terms, one called phenotype and one called genotype. So for example, right here, phenotype is basically what you're showing. So it's like yellow pea versus green earlobe. Or in this case, the phenotype here is all green because they're all green. Or in this case, it's all smooth because S stands for smooth and little s stands for wrinkled. So the phenotype here is smooth, smooth, wrinkled. The genotype is just telling you what alleles you have. So a big S is your genotype. A big S, little s is your genotype. Now your genotype and your phenotype just tells you what your phenotype is going to be. And it's one of those situations. You can't always tell what the genotype is going to be. So if I tell you that you have the phenotype of unattached earlobes, well then you know for certainty, because it's recessive, that it's going to be UU. Now if I tell you that you have the phenotype for the attached earlobes, you don't know what it's going to be. It could be AA or it could be AU because the trait is dominant. This is where things get crazy in genetics because it can get kind of hard. Now, genotype is much better because if you have the genotype, you know what alleles they're going to have. Okay, one last thing before I go on and show you how to actually calculate this stuff through math and actually see how babies are going to come out. So, there's two key terms here, homozygous and heterozygous. Homozygous is when you have the same alleles for both. So if you are YY, AA, UU, GG, you are considered homozygous. Heterozygous just means you have different alleles for both of your alleles. So an example would be like YG, AU, UA, and GY. That's a pretty easy idea to understand. That'll come from practice when you see more and more of that, guys. Two more key terms that all kind of relate to this. There's alleles can be found on chromosomes. So each chromosome has an allele, and when gametes are being made in meiosis, these two alleles separate. And you'll see what that means in this slide here in a minute. Now, independent assortment is a very fancy term. All it means is that each gamete is going to get a different allele. So if you make 4 sperm cells, then each sperm cell is going to have a different allele than the other ones are. So that means alleles are independent of each other. One allele doesn't affect the other. The last one is a key term that I just tossed out here very quickly. It's the word hybrid. It's just a fancy way of saying that it's when you have two people with different alleles and you make babies. So say Mr. Russell finally gets married, and let's say I get married to a blonde woman. Well, my kid would be considered to be a hybrid of my wife and I because my wife and I have very different alleles. Okay, independent assortment. Fancy way. This right here is showing the chromosomes. This is showing the cells before meiosis. So in meiosis, as you guys know, there's 2 steps of division. So what happens is that these alleles move different sides based upon the chromosome, and you end up with different alleles, you end up with different gametes with different alleles. So this one here is different from this one here. This one here is different from this one here. Pretty easy to understand, guys. Just a fancy way of saying that the alleles because the alleles move with the chromosomes. Hey guys, I screwed up the first video, so here's a fix to it, and I'm going to cut this into the video later on. So, I have a sample for a Punnett Square. So here's how you can actually predict traits. So, here's a sample problem. A woman who can roll her tongue has a baby with a man who cannot roll his tongue. the woman is heterozygous for the trait. What is the probability their child can roll his tongue? Okay, well, there's a couple of things in this question you can figure out. First off, it says the woman is heterozygous for the trait. Well, she can roll her tongue. That means that the trait has to be a dominant trait, because if it was a recessive trait, she would not be able to do this. So that means tongue rolling is a dominant trait. Now, if the man, he can't roll his tongue, that means the man is going to be recessive. So we can write that down in this way here. We can say the woman who is heterozygous would have, let's say, big T, little t. So big T is tongue rolling and little t is not tongue rolling. And the male is little t, little t. He would be homozygous for this because of this. And remember, you can figure this out just by how you're going to read the question. Because it says the woman is heterozygous, well, if she's heterozygous, you know that means she has one of each allele. And because she can roll her tongue and she's heterozygous, that means the trait has to be dominant. And because the male cannot roll his tongue, that means that it's going to be a recessive trait. So you can kind of figure out what type of trait it is by being very careful with the problem. And you will see questions like this on your country's exams. That's one question I know it's been on there a couple times. So this is a skill you should probably learn to work up with. So here's how you make a Punnett square. What you do is this. You then put down female over here on the left. You put male up here on the top part. And what you do is you put the alleles here. Now, because of independent assortment, we do know that the alleles go independent, which means this one has to go here and this one will be going here. They won't come together. So you never have a big T, little t here. It'll always be separate alleles. They will always be separate. And the same is true of the male. Always have little t, little t. And what you can do then is you can fill it into these little boxes right here. And what you get is this combination right here, where you have big T, little t, big T, little t, little t, little t, and little t, little t. Well, this gives you the potential alleles of each gamete, because in this case, this would be one sperm cell, this would be the other sperm cell, this would be one egg, and this would be the other egg. And this would give you the total possibilities of babies that this couple could have. Well, now comes the math. Well, you have two cases here where you have big T, little t. Well, because we know it's a dominant trait, we know that here this would be a tongue roller. We're positive on that one. So this part of it here, I'm just going to shade it in. This part right here would be a tongue roller. So you know this is about 50% of the square. And the other situation would have little t, little t. Well, this is a non-tongue roller. This person's not going to be able to roll his tongue. So if you do the math, that gives you a situation of where you have 2 divided by 4 is equal to 50%. Well, that's pretty simple. If you had a situation where all 3 of these were dominant and this was recessive, you would have 1 out of 4 for this possibility, and that would give you 25%. It's pretty easy math, guys. It's not overly difficult, but this is how you could potentially calculate the types of alleles a baby could have. Now, thank you guys for watching the video. Here are some of the key terms that are a part of this section. I decided to add this section in here to help you guys who are asking me about where you can find the key terms. Here are the ideas you should have. I can put them on the Cornell notes, and thank you for watching my overly long video.